Bacteria live all around and inside us every day. Some bacteria help us with tasks like digesting our food, but others are pathogenic, causing disease. Pathogenic bacteria have a parasitic relationship with humans because the infection allows bacteria to reproduce and spread from one host to another, while humans are harmed in the process. Typically, doctors prescribe antibiotics, which are chemical compounds that inhibit or kill specific microbes, to fight off infections from pathogenic bacteria. However, over time, some pathogenic bacteria have developed antibiotic resistance, allowing them to survive exposure to antibiotics. These bacteria are especially difficult to kill, which makes them dangerous for human and ecosystem health. Antibiotic resistance is a genetic trait in bacteria, often stemming from new genes that arise in bacterial populations. These antibiotic resistance genes are initially produced by random genetic mutations. Bacteria that develop antibiotic resistance genes have an increased likelihood of surviving to reproduce in a hostile environment, resulting in strong selective pressure favoring the antibiotic resistance gene. Directional selection results in the antibiotic resistance gene rapidly reaching fixation in the population, producing strains of bacteria that are completely resistant to a specific antibiotic. Some bacteria develop multidrug resistance, such as multidrug resistant Staphylococcus aureus, or MRSA. These bacteria are especially difficult to treat because there are fewer antibiotics that work against them. Bacteria can also take action to spread their antibiotic resistance genes through horizontal gene transfer, with one bacterium transferring its genes to another. This process is one example of bacteria responding to antibiotics, which are abiotic factors in the environment. The most common mechanism for horizontal gene transfer is conjugation, which involves two bacteria forming a physical channel between them to exchange small circles of DNA called plasmids. The receiving bacterium then gains antibiotic resistance traits. The effects of antibiotic resistance extend beyond human health and into the microbiomes of other species, including plants. Many antibiotic resistance genes are carried by bacteria from outside the root epidermis to the interior ground and vascular tissues of the plant. After these genes enter the plant's microbiome, they can reach edible portions of plants like lettuce, carrots, and tomatoes. When people eat these plants, the antibiotic resistance genes can be transferred to humans and pose a health risk. Researchers at UW-Madison are striving to find solutions to antibiotic resistance. Dr. Vatsan-Rahman and his lab are fighting antibiotic resistance by engineering bacteriophages, or viruses that infect and kill bacteria, as an alternate treatment. In mouse models with an otherwise fatal enterobacteria infection, their bacteriophage treatment produced a 100% survival rate. Dr. Rahman also designs mixtures of bacteriophages with different mechanisms to combat new antibiotic resistance genes before they can spread throughout the population. Dr. Tim Bugney and his lab take a different approach, working to discover new antibiotics in marine environments. These antibiotics will provide new treatment paths for bacteria that are resistant to existing antibiotics. As a part of this research, Dr. Bugney has discovered a mycobacterium that can induce production of new antibiotics in other bacterial species, providing an additional mechanism for antibiotic discovery. There are sometimes misleading sources that complicate our understanding of antibiotic resistance. One such source is a Harvard Health Publishing article titled, "Is the Full Course of Antibiotics Full of Baloney?" Doctors will prescribe courses of antibiotics lasting a certain number of days and recommend completing the entire course. This ensures that all bacteria are killed, reducing the potential for developing antibiotic-resistant bacteria. It initially appears that the author questions this guidance, providing a misleading perspective. However, the main text actually states that patients should follow doctor instructions, but that doctors should consider prescribing shorter courses to limit bacterial exposure to antibiotics and reduce selective pressure. Overall, antibiotic resistance is a global health problem, influenced by both biological and sociological factors. Along with biology, factors such as misleading communication, quality of hospital systems, and more all impact the spread of antibiotic resistance. Through research, clear communication, and improvements in antibiotic dosing procedures, we can prevent the development of antibiotic-resistant bacteria and create healthier societies and ecosystems.